Alright, my goal today is to talk about the proximal convoluted tubule to make sure we understand what's going on in that area of the nephron. As a reminder, we can highlight that area here. This is going to be the proximal convoluted tubule. As we think about the nephron and how it's broken down, remember, let's look at this area here. This, of course, is the glomerulus. This is where filtration occurs. We can look at the loop of Henle here. This is focused primarily on water reabsorption. Over in the collecting duct over here, this is going to be primarily acids and bases, but it's this region here in the proximal convoluted tubule that we get most of our reabsorption. In other words, things that get filtered through the kidney, but I don't want to leave the body. I don't want to end up leaving the body. I need to move these things back into the bloodstream from the kidney, and this is where I'm going to do it. When we say most reabsorption, just to give you an idea, this means water. Even though we said that the loop of Henle is a primary site of water, that's where the extra water is added. This is all passive water movement, and we're going to get 67% of our water back through the proximal convoluted tubule. Sodium is also at 67%. Some other things that are important, glucose. 100%. Unless, and you'd recognize this from a previous unit, if I become saturated, then I can't move that back. If blood glucose levels are too high, for example, in a diabetic state, then we will begin to see glucose in the urine. But for a normal, healthy individual, all of the glucose that is filtered through the kidney, through the glomerulus, should be reabsorbed back into the bloodstream in the proximal convoluted tubule. The same goes with amino acids. For building proteins, these are things that we don't want to lose. We exerted effort to get them. They represent energy, they represent building blocks, and we want to make sure we get them back. And then an important ion involved in buffer system, in carbon dioxide regulation, bicarb ion, HCO3-. We're going to get about 80% of that back. Not 100%, because if you remember, in the distal convoluted tubule and collecting duct, that's where the acid-base regulation is going to take place, and I will fine-tune it in that area to make sure that pH balance is normal. So let's go ahead and let's pick a site right here on the proximal convoluted tubule where we're going to zoom in and see if we can identify how we're going to get this stuff back. So let's say that this right here represents the bloodstream, and we're trying to return things from the filtrate back into the blood. So we have a cell as part of the proximal convoluted tubule. That cell is sitting right here, and then this would be the lumen of the nephron over in this area. Now, the most important to all of this, to make it happen, is the sodium-potassium pump. Why? Because that pump creates gradients, and gradients drive movement. So here we want to make sure on the basolateral surface that we're running, this is going to be ATP driven. So there's our pump on the basolateral surface. And of course you'd be familiar with this. We are pumping out sodium and we're pumping in potassium. That is number one. Now, with that, once we set up gradients with the sodium-potassium pump, that gives us gradients. The next step in the process is to use those gradients, specifically the sodium gradient, to do work. Now, we recognize immediately from previous experience that, for example, sodium can run the secondary active transport. As sodium comes in, it will bring glucose. Secondary active transport, sodium going down its concentration gradient, moving glucose against its concentration gradient. We can also use that same sodium gradient, again established by that sodium-potassium pump, to move in amino acids. This is how we're able to collect 100% of glucose, 100% of amino acids, because of secondary active transport. So, secondary active transport with the sodium gradient, established by the sodium-potassium pump in step one. Now, one of the things that we can do with this sodium gradient as well, let's use sodium and let's set sodium up in an antiport exchange this time. Sodium's going to come in just like before, but this time, instead of a symporter like we had in the other one, we are going to take H+, hydrogen ion, and move hydrogen ion out into the lumen. This is interesting. This is going to do a couple of things. Remember, one of the things that we need to reabsorb is the bicarb ion. Well, as we get H+ built up out here, think about the buffer system. What is that going to do? That's going to combine with bicarb, HCO3-, and that's going to generate CO2 and, of course, H2O. Because carbon dioxide is small, nonpolar, non-charged lipophilic, it can freely diffuse across a membrane. We've established a gradient. As we create higher concentrations of carbon dioxide in the lumen of the nephron, the carbon dioxide is going to diffuse readily right into a site of low concentration, right inside this cell in the nephron. Of course, once inside here, CO2 is going to combine with H2O, with water, to create H+ plus HCO3-. Now, this is what makes the whole thing run. I can use right here on the basolateral surface, again, using the sodium gradient, again, using the bicarb gradient that we just made. As bicarb levels increase inside the cell, bicarb this time is going to go down its concentration gradient, taking sodium ion with it. So this gives us another chance to move sodium and keep sodium low inside the cell. This transporter right here, it's got a special name. It's called SLC4A4. It is an electrogenic sodium bicarb cotransporter. It uses the bicarb gradient, CO3- gradient, to move sodium, and it works actually 1 sodium pumped per 3 bicarb ions, which is really interesting. So now if we go back up and we look at what we were trying to move, water, sodium, glucose, amino acids, and bicarb ion, we can see that the whole thing is driven by first establishing gradients, specifically a sodium gradient by running the sodium-potassium pump. Once the gradients are in place, sodium will want to flow into the cell down its electrochemical gradient. We can use that as a secondary active transporter to move both glucose and amino acids. We also use the sodium gradient in an antiport exchange to build a proton gradient in the lumen of the nephron. We then use that proton gradient to combine with bicarb to create CO2. CO2 moves inside the cell, is converted back into bicarb, which we're constantly pumping out, and this allows further sodium to be moved into the blood.